So thank you very much for the kind introduction, and thank you to the organizers for giving me the opportunity to speak to you today about a scientific revolution that is changing our lives, which is known as gene editing. Before we talk about editing genes, though, I would like to remind you of what our genes actually are. So our genes are stored in a macromolecule that is known as deoxyribonucleic acid, or in short, DNA. The DNA is also sometimes referred to as the code of life, simply because it's the molecule that holds the information to make up every single organism on this planet. Interestingly, this code of life is written by just 4 letters, which are A, C, G, and T, and are short for the 4 nucleobases adenine, cytosine, guanine, and thymine. These letters, however, they don't just swim around randomly in your cells like in an alphabet soup, but instead they are tightly ordered in a helix-like structure that consists of 2 complementary DNA strands that are connected by base pairs between the 2 nucleobases adenine and thymine, or cytosine and guanine. So now, this helix structure, as well as the fact that the structure contains the code of life, has been known to us since the 1950s. And ever since, scientists wanted to read the code of life. However, this has been impossible for several decades simply because we did not have the technology to read or sequence DNA. So it took another 40 years for the first, and to date the largest, collaborative bioproject to attempt to sequence the first human genome, and this project is known as the Human Genome Project. So the Human Genome Project started in 1990. It took 13 years and $3 billion to sequence the first human reference genome. And what we learned from the Human Genome Project is that the human code of life consists of 3 billion letters, in which we find roughly 20,000 words, which are our genes, that translate into proteins, which are what eventually makes up your body. What the Human Genome Project also taught us is that it's unacceptable to take 13 years and $3 billion to sequence a single human genome. And this is why around the mid-2000s we see a number of very clever new DNA sequencing technologies appear, collectively known as next-generation sequencing technologies, which today allow us to sequence a whole human genome in just 2 days for $1,000. So we live in the post-genomic era. Today we can sequence genomes on end, genomes of different organisms, genomes of different individuals, genomes of genetic diseases, such as cancers. However, knowing the sequence of a gene or knowing the alterations that we find in certain diseases does not tell us about the function of that gene or that alteration. And this is where the grand challenge lies for the scientists today, which is making functional sense of the identified genomic sequences. And this is where gene editing comes into play. So gene editing is defined as the targeted modification of the genetic material of a living organism. And gene editing is not new. In fact, gene editing has been used for several decades. It's only that until recently, gene editing has been very complicated and very expensive. And so it was reserved to only a few expert laboratories around the planet to edit human genes. This has changed in 2012 with the discovery of a technology that is known as CRISPR-Cas. CRISPR-Cas has basically made it very simple and very cheap to conduct human gene editing experiments, which is why today every scientist on the planet and even non-scientists are able to edit human genomes and other genomes. And because this technology has been so revolutionary, I would like to quickly introduce you to how this technology works. So the CRISPR-Cas system consists of 2 components. One is the Cas nuclease, which is a protein with the ability to cut DNA. And the second one is a so-called guide RNA. So RNA is a molecule that's very similar in structure to DNA, and guide RNA already tells you what this does. It guides the Cas nuclease to its target site in the human genome. So by delivering this Cas9 guide RNA complex into a living human cell, what happens is that this complex starts to scan the full human genome until it finds the complementary target site to the sequence that was programmed into the guide RNA. So we can basically target this complex to any position in the genome. You can imagine that a bit like a genomic zip code. So we tell the Cas nuclease where to go. And as you can see, and I told you before, the Cas nuclease can cut DNA. So what happens is it introduces a cut in each of the two strands of the DNA, which results in a DNA double-strand break. This ability has also earned the CRISPR-Cas system the nickname gene scissors or genomic scissors. And this is precisely how we use this technology in the lab. So by targeting the Cas guide RNA system to a single or multiple loci in the human genome, we can basically decide where we want to cut, and by that we can remove genomic sequences from the genome. You can imagine this much like you would remove a word in your word processor at home at your computer. We can also paste in new sequences into the genome. Again, just how you would paste a new word into your text document, and now the resulting sentence would spell something different. So here in this case, we would have exchanged a thymine for an adenine. And as you remember from the definition that I gave you before, these changes, they happen in live cells, meaning that we can now study the functional consequences of this genomic change in the living cell. So now that you have learned about how gene editing works, let me take the second half of this presentation to give you a few examples of how we use gene editing and how this is changing our lives. So one area that has been taken by storm by especially the CRISPR-Cas system has been basic biomedical research. I would claim that today there's barely any biomedical research lab that is not using CRISPR-Cas to edit human genes to learn about the function of our genes. And since our lab is no different, I would like to give you one example of what we are trying to achieve in our lab here in Halle. So we are interested in the mutations that we find in cancer patients. So as you know, cancer is a genetic disease, and as I've told you, sequencing full genomes has been cheaper and easier than ever. And so today we have access to the sequences of tens of thousands of tumors from different cancer types and different patients. So what we have learned from these sequences is that every cancer type has a specific set of mutations, but also every individual with a certain cancer has a very unique mutation profile. And what we are interested in is to find out how these mutations affect the response of patients to therapy. So we are using gene editing to reversely engineer those mutations into cultured cancer cells, followed by the treatment of those cells with cancer drugs. So this tells us which mutations make the cells resistant or sensitive against a certain drug. The idea here would be now that in the future we can take tumors from patients, sequence the mutation profile, and then, based on our functional knowledge, could categorize the patients into responders to a certain drug or non-responders. This concept is also known as personalized therapy, and it will eventually allow us to increase therapeutic success simply by giving every patient the correct drug. But CRISPR has moved on beyond basic biomedical research already into the clinics. And this is why I would want to give you a few examples for clinical applications here. So one application for which gene editing and CRISPR are already being used is for cancer immunotherapy. So cancer immunotherapy involves the removal of immune cells from a cancer patient, their culture in vitro, and their editing in vitro, where a gene is introduced into those immune cells, which you could imagine like a cancer cell receptor. Now you take those edited cells and reinfuse them into the patient, and now these immune cells, they are armed. They're armed to find and destroy the cancer cells of that patient. So basically you have reprogrammed the patient's own immune system to destroy its own cancer. Another, currently less far developed approach is aimed at curing HIV patients. So here the idea would be to remove blood stem cells from the patient. These are the cells that make your immune cells. And in this case, gene edit them, but not to introduce a new gene, but the opposite, to remove a gene that the HIV virus needs to infect the immune cells. And by that, the cells become resistant to the virus. So now by reimplanting these edited blood stem cells into the patient would produce resistant immune cells, and by that, ideally, could be cured from HIV. And now for the last few minutes of my presentation, I would like to draw your attention to a hypothetical application of gene editing that's considered much more critical than the applications that I had shown to you so far, which is germline editing. So if you take the examples I had just given to you, these all involve removing some of the patient's body cells, editing them ex vivo, and reinfusing them into the patient, meaning only a subset of the cells of the patient are edited. Germline editing is very different in that respect, that here the very early embryo is edited, which means that the edits that are occurring in this early embryo will eventually be present in every single cell of the resulting organism. And that's problematic for a number of reasons. First of all, you remember that I told you that gene editing, and especially the technologies that we're using today, particularly CRISPR-Cas, are less than 10 years old. So we still do not fully understand what the effects are after we introduce those systems into human cells, specifically, especially not in early embryos. So it has actually been reported that in certain cases we see edits that are not on the target site that we want to edit. These are known as off-target edits, and they can have unpredictable consequences for the development of the embryo or the resulting individual. Moreover, as I told you, the edits from the early embryo would appear in every single cell of the resulting individual, and that includes the germ cells of that individual, meaning that these edits would be passed on to future generations, so the children of that individual and their children, including potential off-target edits. So for those reasons, there is currently a broad consensus that germline editing should not be performed at this point in time. However, with increasing safety of the technology and passing years, it is likely that the acceptance also for germline editing will increase. And for this reason, it will be important to establish an international framework to guide and regulate the safe and responsible use of gene editing, including germline editing. So I hope that I could convince you by my talk today that gene editing holds an immense potential to cure some of humanity's most dreadful diseases, and that therefore it would be irresponsible to ban the technology or lock it away. Instead, I think, just like with any new technology, we should approach gene editing with the necessary caution and with respect. Thank you very much.